All right, great. Well, I'd like to thank the organizers for inviting me to talk to you today. And this is the title of my talk, Diversity and Dynamics of the Drosophila Transcriptome. And I was planning on talking to you exclusively about that, but I'll just talk to you about this on the first half of the talk. And then the second half, I'll talk to you about a gene in Drosophila that encodes as many, or twice as many isoforms as there are genes in the entire animal. So it's sort of a whole genome in of itself. Okay, so my lab for the last few years has been involved in the Mod ENCODE project, and the goal of this project, as many of you know, is to identify all the functional elements in the genome, in this case in the Drosophila and the worm genomes. And so there are groups that are identifying all the transcription factor binding sites, groups that are identifying all the chromatin marks, and our group has been involved in trying to identify all the RNAs that are transcribed from the genome. And I'll give you an overview of the data sets that we've generated and some overviews of the results that we've obtained. So the data sets that we've generated, we've published a few of these, but most of them we haven't. So we have a developmental time course that we published a couple of years ago. We now have RNA-seq data on 29 different tissues, 25 cell lines, and then flies that have been treated with about 25 different conditions. So we've treated them with different metals, put them on ice, burned them, put them in coffee, give them Bombay sapphire, and things like that. And then we can ask what happens to the transcriptome after we do that. So as I said, we've generated from this data set about 115 libraries. These are all poly A+ libraries, and nearly 10 billion reads from that, and we've published a number of papers on that. And more recently, we've resequenced all of the same RNAs using a strand-specific total RNA library. So these are ribosomal depleted libraries. And then we've also done some additional ones. So we have about almost 24 billion reads from that particular data set. And we have papers coming out soon, hopefully, describing the rest of these data sets. So now I'll sort of intermingle all of those data sets and just give you some highlights of what we've found. So using all of this data, we've identified about 2,000 new genes that were not previously annotated. And this is a little bit surprising because the fly genome, even when we started this project, was one of the best annotated eukaryotic genomes. And so one of the questions we always get asked is like, how did you find 2,000 new genes? So this is just one example, but it highlights many of the characteristics of these new genes we identified. So the first is, these are the RNA-seq tracks from the developmental time course here. There's 30 time points, and there's this gene I've highlighted in blue here. What you can see is it's really expressed at significant levels only in one of the 30 time points, maybe two of them. So if you never queried that particular time point, you would never see this gene being expressed. The other thing is that many of the gene prediction programs use conservation as a means for identifying genes. And this track down here is conservation, so what you can see is this gene is actually completely non-conserved. So any program that used conservation that would predict these genes would completely miss that. And so this is generally true of the new genes we identified. They're usually restricted in temporal or tissue-specific manner, and they're usually not highly conserved. We also identified a lot of new non-coding RNAs. And here's one here. So we identified about 40 new SNO RNAs. And so they're really good programs that will predict SNO RNAs in the genome. But all of these differ just slightly from all the rules that those programs use. So they were missed computationally. And this is just one example. In this case, this gene called GP210. So in yellow is the RNA-seq tracks from the poly A+ RNA. In blue is the RNA-seq tracks from total RNA. And you can see this very abundant SNO RNA that's encoded in the intron. This is the structure of it. And of these SNO RNAs, we identified about 40 of them, and they're all expressed at different times throughout development. In addition to the SNO RNAs, we were able, using the total RNA sequencing data, to identify all the precursor transcripts for every microRNA encoded in the genome. So these are just showing two examples here. This one, I can't actually see the number from this angle, miR315 or something like that. But this is about a 9 or 10 kb transcript to encode your 22 nucleotide microRNA down here. And again, this is completely invisible in poly A+ data, but in total RNA-seq data, it's abundantly clear. This is another microRNA that was downstream of this gene called HSR omega. So this gene has a phenotype, and the phenotype was always described because when you did poly A plus sequencing, you would see expression of this particular gene. But we now believe that the phenotype is probably because you're disrupting expression of this microRNA that's about 8 kb downstream of this particular gene. And this is just an expression pattern of the microRNA's precursors throughout development. It's advancing slowly for some reason. Okay, so this is just an example of some tissue-specific novel transcribed regions that we identified. So this is a gene desert that's about 30 kb long in Drosophila, and it's riddled with a bunch of repetitive transposable elements, or the remnants of them. And what you can see is on the negative strand going in this direction, there are basically these 9 peaks. And so these are actually 9 copies of a 27 amino acid peptide that are only expressed in testes. And so we see actually a lot of these types of things. We also see a lot of antisense transcripts. So this is just one example. Again, most of these are testes specific. So this is in testes. This is for the Dicer2 gene, which is transcribed from right to left. And you can see this abundant antisense transcript here. And then in the Eve gene, which is a very well-studied gene, we see an antisense transcript going over the enhancer region, again, primarily in testes. Okay, so this is getting into the splicing stuff. So my lab's been interested in splicing, and that's my actual reason why we did all this transcriptome sequencing from my point of view. So we wanted to characterize the splicing component of the transcriptome, and this is just a data slide showing all the new splicing elements that we identified. And the numbers don't really matter, but the bottom line is that we went from about 20% of the genes that encoded alternatively spliced transcripts at the beginning of the project, and we're now somewhere around 62% of genes. So just like in all other higher eukaryotes that have been studied, the majority of genes in Drosophila are alternatively spliced. So the way that we quantitate splicing is we calculate this value we call the percent spliced in, or PSI, and sometimes we use the Greek symbol psi. But the idea is to, in this particular sample, for example, we would have this exon is included for like 98% of the time. And this is using a combination of reads that span the splice junctions as well as the internal exon here. Whereas in this sample, the psi value might be 2, because only 2% of the transcripts express that particular exon. And so then we can look at a change in splicing we call the delta psi of 96, which is just subtracting the psi values from each of these samples. So this is how we look at the absolute value of splicing in a particular sample, and this delta psi is look at the dynamics in splicing between different samples. So if we look at the developmental time course, there's tremendous dynamics in alternative splicing where there's sets of exons that are basically excluded early on and then become more included as time goes on. And this is just one example of a set of 3 exons in the cadherin N gene. There's 3 sets of mutually exclusive exons, and in early development, the very earliest stages of embryogenesis, one of the 2 exons in each of these clusters is included, and as you progress throughout the 24 hours of embryogenesis, you switch completely to the other exon. So even within this particular gene, there are 3 coordinated alternative splicing events. So in addition to dynamics throughout time, we have tissue-specific splicing. So this is looking at all of the splicing events in the fly and their tissue-specific splicing patterns. So again, I can't see the things, but these are like head-specific exons here and whatnot. And we have testes and carcass-specific exons. But one thing we can do is look at the dynamics of alternative splicing. So in this particular analysis, we did compared 3 different things. So the first thing is we compared splicing dynamics between all samples, between the tissues and the time course. So you could have, so what we took was the largest PSI value in any of those samples and the smallest in any of them, and simply subtracted them from one another. So that gave us the greatest range in splicing we could observe. So it could be a particular sample in the developmental time course compared to like testes or something like that. And so when we do that, you see a huge change in dynamics where most of the exons have a very, very big change in splicing. But now if we actually look at just the developmental time course versus the tissues, so the development is in red, there's actually very few changes that occur when we look at the developmental time course over time. Whereas most of the big changes actually are derived from the tissue-specific events. And we think that there's not a huge amount of things that switch dramatically in the developmental time course simply because you have splicing events that might be black and white between two different tissues that when you grind an entire animal up, they sort of weigh one another out, and you can't see the dynamics there. But one thing we can do is if we take the exons that change the most dramatically in the tissues, and we bin them by exons that have delta PSI values of 50% or greater, 25 to 50, and 0 to 25, what we can see is then the conservation of the exons. The exons that change the most dramatically are the most highly conserved in both the exon and the introns flanking them. And so the red is for the really highly switching exons, blue is for the intermediate, and the green is for the ones that change the least. And in addition to the conservation, we see that the exons that change the most dramatically have the greatest tendency to be frame preserving. That is, regardless of whether the exon is included or not, you would not change the reading frame for that particular exon. And again, as you go decreasing amounts of splicing changes, you see decreasing tendency to be frame preserving. So Drosophila is, one of the best things about Drosophila is they have this textbook model of sex determination, which is, involves the sex determination pathway and alternative splicing. And so in this pathway, there are 5 genes that are alternatively spliced one way in males and another way in females. And these are all black and white decisions. And these control all aspects of the sex determination of the fly. So they code both the physical characteristics of the fly as well as the behavior, and they also include regulate dosage compensation. So we were interested in trying to identify additional genes that are sex-specific. And from looking at just the developmental time course data, where we had whole male and female adults that we sequenced separately, we identified something on the order of 800 exons that are sex-specifically spliced. And this is just an example of one of them. In fact, this was previously annotated as a constitutive exon. So in this case, this exon is highly included in males and almost entirely skipped in females. And you can see it's very highly conserved. And in fact, upstream of this exon, there are binding sites for the RNA binding protein, sex lethal. So we think this might actually be a direct target of the sex lethal protein. So we were interested now in looking, since we had all these different tissues, whether there was, in some of the tissues we had, for instance, we had heads from males and females separately. So we were interested in looking more carefully at this in the tissue data. So what we found is if you take the samples from whole animals, male and females, and here we're just using cassette exons, and we look at the correlation between those samples. So we had 3 female and 3 male samples. We see that the females and the males are very similar to one another, but they're very different from each other. Okay? Whereas if we look at the head data, we were really surprised to see that looking at female and male heads, they basically looked identical looking at the splicing levels. Which was very surprising considering there's a lot of literature saying that there's all these sex-specific splicing events. But this is, if we look at actually mRNA levels instead, we can see that there's big differences in the mRNA levels in these. So we were interested in why we're not observing, we see sex-specific splicing in whole animals, but not in heads. And so what we took is all the exons that were sex-specifically spliced in the whole animals, and we looked at where they were spliced in the tissues, and what we can see is the majority of the exons that are male-specific turned out to be included specifically in testes or accessory glands, which are male-specific tissues, and the female-specific exons were predominantly included in ovaries. So we see very little somatic sex-specific splicing, and it turns out that the vast majority of sex-specific splicing is really gonad-specific splicing. Okay, so that's going to be the end of going over the fly transcriptome from the mod encode stuff, and now we're going to work on, talk about data that we've been working on in my lab for a long time on this Drosophila Dscam gene. So this gene encodes 115 exons, and 95 of these are alternatively spliced. And the alternative exons are organized into 4 clusters, where you have the exon 4 cluster has 12 exons, the exon 6 cluster has 48 exons, the exon 9 cluster has 33, and exon 17 has 2. And the exons within each cluster are alternatively spliced in a mutually exclusive manner, meaning that every time you generate a transcript, you include one exon 4, one exon 6, one exon 9, and one exon 17. And all the data that we have to date suggests that the exons within each cluster are spliced independent of the other clusters. So it would theoretically possible then that you'd take 12 times 48 times 33 times 2, and that would give you 38,016 possible isoforms, all of which would encode a full-length protein. Okay, so none of them would change the reading frame. Exons 4, 6, and 9 alter 3 different Ig domains in the extracellular portion of this receptor, and exon 17 encodes the 2 different transmembrane domains that result in targeting the protein to different ends of the cells. So the gene has really interesting biological roles. It plays an important role in axon guidance. Depending on which isoforms are expressed in a particular neuron, that will determine in part the wiring pattern of that particular neuron. And it also appears to play a role in the immune system. So if you delete the gene in hemocytes, which are basically the blood cells of flies, they fail to mount an effective immune response, and different isoforms of this bind to different bacteria in a bacteria-dependent manner and isoform-dependent manner. So we're interested in many aspects of the splicing of this gene. So we want to know, for instance, how many of these theoretical isoforms are actually expressed, what are the mechanisms by which splicing is mutually exclusive, and then how is the splicing of this gene controlled. So one way that we've been getting at how many isoforms are expressed is to use this method we developed called triple read sequencing, where we basically have focused on just the exon 4, 6, and 9 regions of the gene. So we amplify using RT-PCR from exon 3 to exon 10, and then the problem is exons 7 and 8 are about a kb long, so this whole fragment is now 2 kb, which is really too large to cluster on an Illumina sequencer and sequence it effectively. So what we do is we circularize this and then do inverse PCR, and that gets rid of the large exons, and this gets down to about an 800 base pair fragment, which we can actually cluster. And then we use primers that anneal to the constitutive exons, and we sequence a few bases in, and in between each step we strip the sequencing product off and anneal the new primer and whatnot. So we can, from every cluster we get 3 different reads, so we can tell which exon 4, 6, and 9 isoforms are expressed or present in each cluster. So doing that, this is just one experiment where we had about 3 million independent clusters, and this is the frequency of which we observed each individual exon in those. So we observed all but about 4 of the exons included in this particular experiment, and since we just focused on exons 4, 6, and 9 and not exon 17, there were only 19,000 potential isoforms, and we basically observed 92% of them in this single experiment. There's a huge diversity and dynamic range in which they're expressed. So there was one isoform we observed 42,000 times, and many of them we only observed once. So the data so far suggests that most, if not all, of these potential isoforms are expressed. And actually, Dietmar Schmucker's lab recently published a paper where they had a very similar approach and came to essentially the same conclusion. So we think that most, if not all, of these isoforms are actually expressed. So one question from a mechanistic point of view is like, how is it that you can include only one of the exons in each of these array of exons? Okay, so I don't have time to go into it, but there's many mechanisms that people have uncovered for how mutually exclusive splicing works in other genes, and these are cases where you have only 2 exons. But none of them can work for Dscam for various reasons. Certainly when you have 48 exons in a row, none of the mechanisms that are known to date can explain why why it's mutually exclusive. So a few years ago, we uncovered part of the mechanism, and this was by comparative genomics, where we simply aligned the Dscam sequence from 16 different arthropods, and we identified these really highly conserved sites. The first one is located between exon 5 and the first exon 6 variant, of which there's 48. And this is the consensus sequence of it here. And in fact, this is the most highly conserved sequence in the entire gene. This is more conserved than the constitutive exons, and it's sitting in the middle of an intron. And this is conserved over about 500 million years of evolution. So now, upstream of every alternative exon, there was a shorter sequence that was less conserved than the docking site, but still conserved. And we noted that they were similar to one another, so you could draw this consensus sequence, and we called these things the selector sequences. And what became apparent for how these things were working is that if you flip this around upside down, it will base pairs with this docking site. So what would be going on is you have exon 5 here, and this would be the selector sequence, or the docking site in red. Then you'd have the selector sequence for exon 6.1, and that would base pair there. And the binding of each of these selector sequences prevents the binding of the 47 other selector sequences. So when one of these binds, it's mutually exclusive with the binding of all the other ones. And one thing that supports these models is the fact that this docking site is almost invariant, which would be predicted because if you mutate any of these nucleotides, it would impact the binding of almost every selector sequence and screw the entire system up. Whereas the selector sequences themselves can be more variable because you can mutate one and it'll only impact the splicing of that one particular exon. So we've actually done a bunch of experiments now where we can mutate any of these elements, and if we increase the base pairing potential of any selector sequence, that increases the splicing inclusion of that particular exon, and if we decrease the splicing or the base pairing potential of a particular selector sequence, that actually decreases the base pairing potential and the inclusion of that particular exon. So we've also done some RNAi screens where we've identified a protein called HRP36, which binds to and represses all these exons. So we think the mechanism by which the mutually exclusive splicing works is that all the exons are repressed by this HRP36 protein. We get base pairing interactions between the selector sequence and the docking site. Something magical happens, and we think that the exon immediately downstream of this base pairing is somehow stripped off of HRP36, and this another protein, perhaps an SR protein, comes in and will base pair there, and then you can get splicing of that exon. And because the remaining exons downstream are still repressed by this protein, then you can only splice down to exon 7 at the very end. Okay, so one of the things that we've really been interested in is looking at when and where the individual isoforms are expressed. And this also will get into one of the interesting aspects of the regulation of splicing for this particular gene. So one way that you could go about looking at where and when different isoforms are expressed is you could do like single cell RT-PCR, but as we know that's like technically very challenging, especially when you have 38,000 potential isoforms to look at. So what we decided to do was focus on this exon 4 cluster, which has 12 exons, and develop tools that we could look at these in single cells in the animal. So in collaboration with Larry Zapersky's lab, we built a series of splicing reporters for the exon 4 cluster, where downstream of exon 5, we've used Gal4 transcription factor, and between them we put a 2A self-cleaving peptide sequence. So when an exon would be included, that was in frame with Gal4, we would cleave it right here, and that would just liberate Gal4 so that it could then go on and act like a transcription factor, and we didn't have to worry about what was fused upstream of it. So then to look at a specific exon, what we would do in that reporter is leave that exon wild type, and we actually inserted a single nucleotide in the 11 other exons, so any of them all the rest of them would be out of frame. And we'd only get Gal4 produced when that particular exon was included that was not mutated. We ended up building 14 different reporters, one for every single exon, and 2 controls, one where none of the exons had a frame shift, and one where all the exons had a frame shift. And all these reporters were then homologously recombined into the endogenous locus, so one allele was the wild type gene, and the other one was the construct, or was the endogenous gene carrying the recombined reporter. And we wanted to do this so that we would have the endogenous transcription pattern of this particular gene. So then, by using Gal4, we could do 2 things. One, we could amplify the signal that's, from expressing this, and then we could hook up with all the powerful Gal4 tools that exist in Fly. So for instance, we can put this reporter into a fly that has UAS sites driving the expression of myristylated GFP, and that would allow us to label the entire cell body of neurons expressing a particular exon. Okay. So here we go. So we've looked at several different neuron subtypes. So the first one was in the mushroom body. So these have these Kenyan cells that express their neurons or their axon projections down here. They bifurcate here and they make these two lobes. So this would be on like the left side of the fly, and you're looking at it from the fly would be facing you. What's shown here in blue, this is the negative control. So white is the expression of the GFP reporter. So in the negative control, where there's a frame shift for all 12 exons, we basically see nothing. And the white over here, this is the positive control, where all the exons, none of the exons have a frame shift. So every single exon should light up. And so what you can see is from the mushroom body is basically all the neurons here are expressing the Dscam reporter. Now, if we look at individual exons, this is just 4 of them. So the 2 rows shown here are for 2 different individual animals expressing the same reporter, so you can see the variability or lack thereof. And so for exon 4.1, for instance, we see sort of a moderate number of neurons expressing the reporter. Exon 4.2, we see hardly any, maybe 2 in each particular animal. 4.9 is a little bit more, and exon 4.12 is very high. And so most of the neurons express exon 4.2. And we have data for the other 8 exons, but I just didn't show them here. So we see a large number, or quite a variety in the distribution of the number of neurons expressing each particular exon, but the relative number in every single individual for a particular exon is the same. Okay. So one thing we can't do, because the mushroom body system is too complex, is we can't pinpoint the same neuron from animal to animal to see if the individual neurons are expressing the same isoform in individual animals. But we can do that with other systems, for instance, the DA neurons in the fly. So these are some peripheral neurons. If you take the animal from the anterior to posterior, and you basically, as a larvae, you slice it down the middle and you fillet it. There are 7 rows of 3 neurons on each side of the fly, and you can pinpoint every single neuron from animal to animal. So we can actually count, how many animals express exon 4.2 in that particular neuron. And so what we did here, instead of using myristylated GFP to light up the entire cell body, we used Gal4 driving histone GFP. And so when the nucleus turned white, we can actually see, say that's expression of the particular reporter, and this would be off. Okay, so these are the 2 controls. So these are heat map representations of this. So each row is one of those 42 neurons, and each column is a different individual. So we have 6 to 8 individuals for each reporter here. So what you can see with the negative control is that everything's off. There's 2 neurons in one animal that had something on, and the positive control is basically on in everything. And looking at 3 different exons, again, we've done it for all 12, you can see a different diversity of exons are actually expressing each of the, or different diversity of neurons are expressing them. So for instance, exon 4.2 is on in almost every neuron in every animal, whereas exon 4.1 is almost off in almost every neuron. Okay. So the bottom line of what I want to show you here is that we've uncovered from these experiments this idea of probabilistic splicing. So what I mean is that in mushroom body neurons, there's always about 50% of the neurons that express exon 4.1, and always about almost none of the neurons express exon 4.2. But in a different subclass of neurons, this is switched, where exon 4.1 is basically off in almost everything, and exon 4.2 is on. So the probability of splicing exists in every single neuron within a class, but this can differ between classes. And we actually think that the repertoire of RNA binding proteins that are expressed in each subclass of neurons determines the probability of every exon in each of these subclasses. And we sort of equate this as to these 12-sided die from Dungeons and Dragons when we were kids, where they would all be weighted in different ways depending on which neural subtype you are. Okay, so rather than summarizing this, I will just thank the people that did the work. So the mod encode stuff was a large group of collaborators that I don't have time to mention everyone, except Sue Selnicker was the main PI of the mod encode transcriptome project. And with the Dscam stuff with the reporters, Satoru Mura was a graduate student, and that was all done in collaboration with Larry Zapersky's lab. And I have to thank NIH for money. So thanks a lot for your attention.
Any questions for the speaker? I have one question.
Yeah.
So these probabilities of inclusion of the exons will be conditional on the other proteins that may be expressed, right? RNA binding proteins.
Right.
Can we use the data to already try to characterize motif finding, or it's?
So I don't think we can use this data to determine what those proteins might be, but we're doing experiments in the lab where we've done RNAi screens in tissue culture cells, and we can, even by changing 2 proteins, we can dramatically switch the repertoire of which isoforms are actually expressed. So we're now going into the animals and determining if those same proteins play a role in the animal.
Cool. And how noisy is the splicing? I mean, does it always has to be determined, or you can also have multiple isoforms at the same time?
So we don't yet know how many isoforms are expressed in an individual cell. We do know that it can change over time. So one of the slides I skipped over was doing live imaging at early and late larval stages, and we can see it change. So sometimes it's on early and it goes off, and we see the reciprocal happens as well.
Thank you.
What proportion of the new, if you know, what proportion of the new transcripts that you talked about in the first half of your talk are either transposons, or at least they're driven by LTRs or something like that? And if it's low, then what do you think is, how do these new transcripts get there?
Yeah, so we think there's very few of them because we put the transposon and repeat-based transcripts into a separate bin and analyze them separately. So these are like unique, most of them are protein coding. So there's not a huge number of non-coding RNAs, at least in the sense of like what you think of as link RNAs in humans. There's not a large number of those in Drosophila.
I have a question about the calculation of a PSI. So before calculating PSI, did you normalize the RNA-seq data, or did you do any sort of data pre-processing?
So the way we do it is we basically align the reads. We have a minimum cutoff. So I think in this case we were doing like a minimum of 20 reads that were informative one way or the other, and for a particular splicing event. So these are all would be like the flanking constitutive exons and the alternative exons. So we're not looking at reads outside of that region. And then we'd have a minimum of 20 reads for that particular splicing event, and then we simply calculate a ratio of reads that are inclusion versus skipping.
I see. So you are not comparing the splicing events, the percent of the PSI at that exon. You didn't compare that with other exons?
Not with other exons. So we compare that particular exon
itself
in sample A to that particular exon in all the other samples.
I see. Thank you.
Mm-hmm.
That was a beautiful talk.
Thank you.
Do you know, or would you speculate on why different isoforms are expressed in different subpopulations of neurons, and what would potentially happen to those particular neurons if you messed with it?
So with regard to the Dscam, the way that Dscam works is that so it engages in isoform-specific homotypic binding events, and so if two neurons express the same isoform and they interact with one another, they repel one another, which seems counterintuitive, but all the neurobiologists say this is the way it works. So if you express the same isoform, you repel. So the way that neurons interact with one another is they have to express different isoforms. And so the thought is that the reason that you can make 38,000 potential isoforms is that the probability that neuron A will express a different isoform from neuron B is basically 1. And so and the other thing it does is every neuron itself by definition expresses the same isoform, and it's also involved in branching of the neurons. So where they branch, the idea is that there's interactions going on there that cause the two ends of the neuron to go away from one another.
I have a very quick question. So the cell is spending a tremendous amount of energy to produce all these splice variants, distributed along what looks like a log normal curve. Or do you think they're often translated, usually not translated? Do they have some kinds of partners that they interact with? In other words, what's happening with that? You have to build it, break it down, recycle. It costs energy. What's going on? Do you have any ideas?
Oh. Yeah, we haven't done mass spec on the 38,000 isoforms. So I don't know. I mean, we don't have any reason to believe that they're not translating them when they're being made. It doesn't mean that they are. But when I do this, when I say we see all these different isoforms, that's like from grinding up a fly head. So there's 250,000 neurons in a fly head. So each neuron could be expressing 5 forms, and it could be that just a lot of them express one particular form, and very few of them express another form, and that could be the diversity that we're seeing there. So we haven't gone and done it at the single cell level, where we do the sequencing to look at how many isoforms are expressed in a single cell.
Okay, let's thank the speaker again.
Thank you.